The National Small Grains Collection, which is part of the National Plant Germplasm System operated by USDA-Agricultural Research Service. We're located here in Aberdeen. So for each of the crops, wheat, barley, oat, rye, triticale, and rice, we have cultivated and wild species for each of those crops. Roughly, the wheat collection is the largest at over 60,000 accessions. Barley is around 30,000. Oat is around 20,000. Rice is around 19,000. Rye is around 2,000. Triticale is around 2,000. Aegilops is around 2,000 accessions that we hold. These are worldwide collections, and I think you could go down each crop and say wherever these crops are grown in the world, they're represented in the collections here at Aberdeen. So pretty diverse, fairly complete. The National Small Grains Collection, history-wise, best I can tell, has its origins in about 1948, which assembled separate crop collections in Beltsville, Maryland. Before that time, there were separate crop collections for wheat, barley, oat, rye, and triticale, and rice, I should say also. Each one of these crops had USDA serial investigators that sort of managed the different crop collections of germplasm. And they established their collections in different ways. There's a lot of overlap with the CI numbers also. Serial investigation numbers can be confusing because each one overlaps the other. So for wheat, we call them CITR for CI triticum, and CIHO for hordeum, for barley, etc., etc. Each one has their own group. Those crop collections were then brought together into the National Small Grains Collection. The collection was held physically there in Beltsville until 1988, when this facility here was constructed, completed. The collection was physically moved here to Aberdeen at that time. Now, prior to it being moved here, Aberdeen has been involved with the grow-outs and evaluation of these small grain crops since the early part of the 20th century. So USDA scientists during those years, they would spend part of their summer season here in Aberdeen or other western locations. So the grow-outs were mostly done here in Aberdeen for the spring crops. The winter crops were grown down in Arizona quite a bit during that same period of time. So there were a lot of interactions with USDA scientists during the summer seasons here in Aberdeen over many decades. We're standing in the winter habit nursery that was planted mid-September 2023. It's winter wheat materials. We also plant winter rye in this grow out also. This particular grow out only has 300 or so accessions growing. Each grow out nursery is anywhere from a few hundred to even a few thousand accessions. Depends on the season or each particular year. We try and level things out a bit, but we do have some big grow outs and some small grow outs. This is again the winter planting. We have a separate spring planting, which is larger than this one. In the case of wheat, rye, triticale, these crops we plant about 8 grams of seed, which would be close to 200 seeds. The basic setup for the fields is that we plant a single row of the accession. Then we have a row next to it that is another crop, in this case, the winter barley. So we separate each accession with a guard row. Another part of the setup of the field is that we plant through and then turn around and come back. So into the next range of materials, the guard row and the accession row are flipped. So as we drive through the field, we're not dragging seed from one accession into the next accession. We're dragging it into the guard row, if that does drag. We treat wheat, triticale, barley, and oat as a self-pollinated crop, which means we will plant one accession next to another accession. Rye is an outcrossing crop. So we're using distance in the field to separate them and hopefully minimize cross-pollination. The summer harvest season every year is a little different, but we're now into July. It will be late July or perhaps early August when these are actually harvested or threshed. We're using one row binders that cut down the row and wrap string around it. So they're hauled to the stationary thresher, which is a machine that we call the Vogel thresher, and the seed is collected. And that's taken into the building. So in this nursery, we're growing spring oat, spring barley, spring wheat, triticale, and rye. We're set up with sort of alleyways between different ranges of materials. In the alleyways, we plant with a crop like spring wheat or something that will just cut down, get out of the way before harvest time. But the main purpose is to keep for weed control. In the case of oat, we plant spring wheat between the different accessions of oat, right, as a guard. And again, the guards and the accessions are flipped as you go from range to range. So seed selected for regeneration is based on sort of a cycle. In the case of, let's say, for example, the case of wheat, we go on a 17-year cycle. So we'll look for seed that was last grown 17 years ago and set that up. Also look at the same time for materials that are low viability and low seed number or seed on hand. Those are the three factors. This was planted in mid-April, I believe. Harvest will be in the month of August sometime, probably extending over a couple, 3 different weeks as they mature. Basically every day as the harvest season progresses, the materials are hauled into the building and put into one of the cold storage rooms and held there until they are cleaned and can be put on the cold storage shelf. The viability test would follow that after it's been on the shelf. If it's a new accession or an accession that's never been studied before, we'll make sure we have enough seed set aside to send to NLGRP for backup. So a good harvested row from material like this, when it's threshed and weighed, will give us anywhere from 200 to 400 grams of seed, which is more than sufficient to put into the cold storage as the active inventory and will last generally the whole life of the seed, and we'll still have plenty of seed inventory on hand. For something like a facultative type where you can't get everything to grow well in one location, like here at Aberdeen, Parlier, California in the Central Valley is good because it's planted in late November, goes through the winter season. The climate is mild enough that it won't winter kill anything, but it is cold enough that it will vernalize, and then you should be able to harvest everything that's in this row. We have 3 cold storage rooms, this one, and there's 2 more across the hallway. This is medium-term storage. We store at +5 centigrade. Humidity is less than 25%, So cool and dry. We can keep seed in good condition for 15 to 25 years, depending on the species. Everything is in order with PI numbers. We also use some of this old CI numbering system too in our accessioning. It's all stored on the shelves in paper envelopes, so everything collaborates to the room conditions. Storage, we have it's just short of 150,000 accessions. The germination testing we do here is mostly for our internal purposes. We want to just monitor the health of the seed. We try and get an initial test within the first year after harvest, and if we have resources, we'll test to 5 and 10 years later. We put 50 seeds in a germination box with blotter paper. It's put into a germination chamber at, there's a couple of different temperatures that we use, but it's a week-long test. At the end of the week-long test, germination percent is calculated and recorded. So if it's new seed, we would certainly expect 90% or greater for any of the crops that we have. After 5 and 10 years, again, for wheat, it might drop a little, but hopefully not a lot. It generally doesn't drop a lot over that period of time. Barley and oat really hold their viability or germination levels for quite a long time. And so after 5 or 10 years, they're still usually in the 90s. So, but there may be an odd accession, or maybe that is perhaps vulnerable to cracking or something in the threshing process, or some other reason. Maybe it was a little green at harvest, who knows? That will give us a red flag. So we have a flow cytometry instrument that we use for ploidy analysis. I think an overall goal using that machine, if we have the resources and time, would be to evaluate pretty much all accessions in the collection for ploidy level. Our main reason or impetus for getting the machine in the first place was to look at wheat land races, because wheat land races growing in the Middle East and Western Asia are frequently a mixture of a durum wheat, which is 4X, and a bread wheat, which is 6X. And most of the time, the morphology will show the difference between the two, but there are examples where the morphology is so similar that you can't tell by just looking at them in the field or in a picture of any kind. So it's necessary to actually go deeper into the cell and count the chromosomes. Eventually, we'd like to look at all of the other crops and all of the wild species, because wild species identification itself can be a problematic thing, and ploidy level can help a lot in that situation. So that's why we do it. We are invested big in imaging. We use flatbed scanners for the most part, capturing images of the kernels that are stored, images of the spikes, that would be barley, wheat, triticale, and panicles, that would be oat and rice, so that we can capture shapes, sizes, and colors. And in the case of the spikes and panicles, we do use color bars and a little measuring device in the image, so you can actually see some scale. But all of those images, in many cases, multiple images, are uploaded to the GRIN Global database and are visible online for use by us as curators and managers of the collection, but also for users who are looking for germplasm. We distribute seed on request to scientists, plant breeders, educators who are improving the crops or studying the crops diversity and so forth, and educators who are demonstrating their use in college level courses. Requests come in through the GRIN Global database online, although I do get email requests. We distribute anywhere from 20 to 30 or 40,000 accession samples each year in 400 or so different requests. 2/3, 3/4 of those are within the U.S. The other third are outside the country.